that kind introduction. I'm very grateful to the Foundation for having included me in this year's symposium because I've had a wonderful week. And I also want to thank a number of the people who made my life easier, especially Alan, who initiated the correspondence, and Ascension, who was really incredibly efficient in helping to organize it. And both of them really for arranging to fix a broken tooth that I came to Geneva with, and that would have made my week really miserable if they hadn't done it. But as it turned out, it's in better shape than when my dentist in Cambridge had actually installed it, so you know. And everybody has been really wonderfully hospitable, so I want to thank everyone. Now, you may have noticed that I put up these pictures not because they're dead white European males, but because they have something in common. And by now, since you're all very smart, you've figured out that they all died young. And they all died of infectious disease. And they died of infections that probably today they would not have died of. Okay? In fact, for many of them, we know that we could have saved them today. And we like to think of those days as over. But in fact, even today, we have over 100 million people who have active cases of tuberculosis. And there are about 2 million deaths a year. Now, we think of tuberculosis as a sort of disease of the third world and not our problem. But in fact, it is becoming a problem even in the West, okay? Because there's a synergy between TB and HIV. And when people are immunosuppressed, they're more susceptible to catching TB. And so it's an increasing problem worldwide. And of course, worldwide, millions of people, regardless of the country, are dying of it. But even in the West, there's a problem with this bacterium called Staph aureus. Aureus, as you know, means gold. And Staph aureus is called that because of this gold pigmentation. This is an organism that likes to invade wounds. And so it's a very common infection after surgery, after operations in hospitals. And when you look at Staph aureus and its response to penicillin, you notice that in 1940, this shows what fraction of strains were resistant. Essentially, all of Staph aureus was sensitive to penicillin. But very quickly in the hospitals, resistance started to emerge, and with a lag of about 2 decades, resistance started to emerge in the community, in the larger community outside hospitals. So today, if you look at Staph aureus, there are other drugs, antibiotics that can be used against Staph aureus. But there are now strains of Staph aureus that are resistant to a wide variety of antibiotics. And these are called MRSA, methicillin-resistant Staph aureus. And they kill more than 25,000 people a year in just Europe alone. Okay? So it's clear that we need antibiotics. Infectious diseases are something we had thought we had conquered, but really we had just won a battle, not a war. Okay? And they're emerging. So I'd like to take you through a brief survey of how antibiotics were discovered. In the modern era, the person who thought of the idea of antibiotics was this man, Paul Ehrlich, a great German scientist. And he had the idea that maybe you can have a magic bullet that attacks bacteria but doesn't attack the patient. And the reason that he thought of that was because he noticed that if you had cells and you stained them with a dye, then there were certain dyes that would be preferentially absorbed by bacteria. The bacteria are shown here as these small rod-like objects. And then these large cells are human cells. And you notice that the dye is not concentrated in the human cells, but it's highly concentrated in these bacteria. And so he thought, well, what if the dye were poisonous? Then the bacteria would preferentially take up this poison and they would die, but the human cells, because they didn't take it up, might survive. And so he started surveying for compounds. He was not terribly successful, although he came out with an arsenic compound that was used for syphilis, but it turned out to be rather toxic. But somebody who was influenced by Ehrlich was this man, Gerhard Domack, who worked for the Bayer Chemical Company and therefore had access to thousands of dyes. And what he did was he systematically surveyed thousands of dyes in a very similar way that we would now survey large libraries of compounds. And he came out with something called Prontosil, which was a red dye, and it turned out to be the first highly effective drug against fatal diseases, for instance, caused by streptococcus and pneumococcus. And in those days, if you got cut or you got infected by playing in the dirt, it turned out that this infection could travel down your arm and eventually kill you. And often you would have to be amputated as soon as the infection was discovered. And so one of the things that he did was to actually save his own daughter from having her finger amputated by giving her Prontosil when it was just becoming clear that it was efficacious. Now, he was unlucky in some other ways. One was that the dye, which was red Prontosil, about a year later, it was found that the way it worked was that it was broken down in the body into a compound called sulfanilamide, which was colorless but had been previously known. And so the patent on Prontosil was void, and neither he nor, the Bayer Chemical Company made a lot of money by making sulfanilamide, but not due to the patent. The other unfortunate thing is, because it was the forerunner of all sulfa drugs and cured really millions of people, he was awarded the Nobel Prize for Medicine, I believe around 1940 or so. The trouble was that the Nobel Peace Prize in Oslo had been awarded to a German pacifist who was an anti-Nazi. And this infuriated Hitler, and Hitler didn't make the distinction between Norway and Sweden, even though Norway, as we all know from Quisling, eventually became a German ally and Sweden remained neutral, but somehow Hitler was confused in many ways. But one of the things he did was to ban Germans from receiving the Nobel Prize. And so Domack had to decline. But after the war, the Nobel Foundation kindly invited him to the next ceremony, and they gave him the certificate and the medal. And they told him, unfortunately, we've returned the prize money to the general funds, so you can't have it. So, but on the other hand, he's a great man who will be remembered forever for having discovered sulfa drugs. Now, sulfa drugs are based on synthetic chemicals, and around the time sulfa drugs came into vogue, this man, Alexander Fleming, who was a Scottish microbiologist, who, after he graduated, his mother said, "My son, it's better if you go off to London to work, because if you stay in Glasgow, you'll have to compete with other Scots. But in London, you will only have to compete with the English." Anyway, so he went off to London. He followed his mother's advice, and he noticed that a plate that got accidentally contaminated by a mold seemed to secrete something that prevented the growth of these bacteria. If you notice, these colonies get smaller as you get closer to this mold spore. So he correctly inferred that the mold was secreting some compound that was lethal to the bacteria, and he called it penicillin. Now, he didn't actually develop penicillin further, but it turned out more soldiers were dying of sepsis than were dying of bullets in World War II. And so the British government launched a major effort to see if they could actually come up with a useful antibiotic. And a group at Oxford, led by Florian Chain, managed to make large amounts of penicillin. But large is relative. Penicillin was so precious that when they wanted to see if it worked, they would give it to a patient and then extract it from the urine of the patient to then preserve the penicillin so that they could re-inject it. And unfortunately, the first patient they gave it to was a male man, and as soon as the fever subsided, they stopped the treatment because penicillin was so precious. And the man got a relapse and died. And so there's a famous saying in medicine, which is, the treatment was a success, but the patient died. Okay, and that's what it comes from. Now, penicillin was dramatically successful and saved, a huge number of lives and continues to do so. But it's not effective against all types of bacteria. Bacteria are classified according to whether they can absorb a stain by a Danish microbiologist named Gram. And they're called gram-positive if they stain with the Gram stain, or gram-negative. And penicillin is not effective against gram-negative bacteria because they have a more complicated surface envelope which prevents penicillin from going in. But there are other bacteria which could be effective against gram-negative bacteria. But there's a class of bacteria called mycobacteria, of which Mycobacterium TB and Mycobacterium leprae, which causes leprosy, are the two most known pathogens. And notice that they have an even more complicated sheath, and many antibiotics are not useful against TB. And TB, as you know, was a worldwide scourge, and many of those people that I showed you on the first slide actually had died of TB. And when Fleming came to lecture in New York, one of the people who heard him was this man, Selman Wakeman, who was a microbiologist at Rutgers. And when he heard that, he was a microbiologist who worked on soil bacteria. And he went back and said, look, if Fleming can find something useful out of a mere mold, we can surely do better with all these varieties of bacteria. And he persuaded his student, Albert Schatz, to start looking for things. And Schatz, after many years of painstaking work, came up with a compound which turned out to be useful. And he first noticed that this compound was actually effective against a wide variety of bacteria. So this is the organism Streptomyces griseus, which is secreting the compound. And notice that it prevents the growth of many different types of bacteria. Otherwise, they would grow all the way up to here, but you can see the growth is inhibited. And then the most remarkable thing is that he showed that increasing concentrations of this, whatever this compound was secreting, would also inhibit the growth of the bacterium that causes tuberculosis. And so it became the first useful antibiotic against TB, for which Wakeman was awarded the Nobel Prize without Schatz, because soon after thing became a success, he slowly, in all of his talks, stopped mentioning Schatz's name. And so much so that Schatz eventually had to sue Wakeman for credit and won the lawsuit. And both Rutgers and Wakeman had to then agree that Schatz was a co-discoverer of streptomycin. And but it was terrible because, you know, this, and it's unfortunately true today too, that if you pick a fight with your professor, you know, if you look for a job, then, you know, people are going to ask your professor, you know, how is this guy? And it's a terrible thing. And I think the lesson is, you know, that professors really need to be gracious. They have absolutely nothing to lose by giving credit where it's due. In fact, if you give credit to your people, you look better for it, you know, and you don't lose any credit. And that's a lesson that we need to learn. Now, many antibiotics, that led people to do a systematic survey, and many of these antibiotics come from a single genus, Streptomyces, which has many species of bacteria that grow in the soil. And that led to the discovery of many different antibiotics. And it turned out that all of these classes of antibiotics work by blocking protein production in bacteria. So that leads, of course, to the question of what are proteins and why are they important? In other words, why is it that if you block protein production, these bacteria would die? And how are they made? Because you need to understand how they're made in order to understand how they're stopped from being made, okay? Now, that, of course, leads to the theme of this symposium, which is the architecture of molecules, because antibiotics are molecules, and they're also made by a molecule. And so I'm going to start showing you molecules. And molecules can be shown in many different ways. And Rod McKinnon, I was afraid he was going to steal my whole story, but fortunately not. But he pointed out there are different ways of showing you molecules. And what I'd like to tell you is that there are different ways of showing people, okay? So as you know, I'm an American citizen, but I want to be bipartisan here by showing a Republican and a Democrat. Incidentally, I've been put up by the Wright Foundation in the same hotel where this man stayed, okay? So anyway, this is a very realistic picture of people, okay? It's a photograph, and that's what they look like. But now you can simplify this and still be able to recognize the person. And so that's shown here, okay? So that's what we call a cartoon. And in fact, molecular structure people also refer to their diagrams as cartoons based on this idea that you can have a highly simplified representation and still be recognizable. But you can simplify it even further, and what you get is a symbolic representation. This is what you would call an icon or an iconic or symbolic representation, where you've essentially captured the essence of the person or of the molecule, okay? So it looks nothing like the molecule, but captures some critical essence of the molecule. And that's sort of shown here, okay? And this is, if you are familiar with Doonesbury, who's a very famous cartoonist in America, this is Bill Clinton, because when he was a president, he could never make up his mind about this or that, and so he was called a waffle. A waffle is, you know, can go either this way or that way, and it's the same, okay? And that's a metaphor for someone who can't make up their mind. And this is based on a Texan saying called "all hat, no cattle," okay? So that's George Bush. So now that I've shown you different ways of showing people, you can also see different ways of representing molecules. Now, molecules are basically structures consisting of atoms that are linked in a very specific way with bonds. So you can show them in a very detailed way by having spheres that represent the atoms, and the size of the sphere has something to do with the actual radius of the atom. And so you'd have a collection of balls, okay? And of course, these actually are slightly shrunk. Rod in his talk showed you what it would really look like if you had the full radius of the atom, and this would all be completely filled up in a solid way. The other way is to show atoms as points that are connected via sticks to the atom that they're chemically bonded with. And so this is the famous picture of Crick and Watson with a stick model of DNA that they had built up using rods and sheets from the workshop. Now here is a schematic representation of DNA. Now notice that here you can sort of see something about the architecture. Here looks like a mess, and you really have to look at it carefully. But in this schematic representation, you've captured the essence of the molecule. And what's the essence of the molecule? The essence of the molecule is that it consists of two strands that are woven around each other in a double helix. And it consists of 4 types of building blocks. And each type of building block can only pair with a specific other type of building block, okay? So that means the building block called G can only pair with C. And they're shown schematically as these pegs with little grooves and notches. But of course, in reality, that's not what they are, but that's the schematic to tell you about specificity and the types of molecules. So this is DNA, which is what our genes are made of. But remember, antibiotics work by stopping cells, bacteria from making proteins. So what are proteins? Well, here are 3 types of proteins. Proteins are long chains. They're polymers of amino acids, which are different kinds of chemical building blocks. And they can fold up into different shapes. So just to give you an idea of variety, this is collagen, which is a long filamentous molecule. And this molecule makes up our skin and connective tissue. And without it, we would have no form. We would collapse into a blob, or we would just be drooped around our skeleton. And so that's what gives us structure. This is hemoglobin, which is one of the first proteins whose structure was solved by Max Perutz, who founded the institute where I worked. He was actually Francis Crick's PhD supervisor. And this is hemoglobin, and what it does is carry oxygen from our lungs to our muscles where it's needed. And this is rhodopsin, which sits in the membrane or envelope. You saw many of these types of proteins in Rod McKinnon's talk, but this particular protein sits in the membrane and detects light. So it's in a membrane, in the membranes of our eyes, and it's the reason that you can see the screen, because rhodopsin in your eyes is detecting light and sending signals indirectly through your brain. So how are these proteins made? So proteins carry out the thousands of functions in the cell. Antibodies are proteins, for instance, the things that we use to fight infections. All of the channels that Rod McKinnon talked about on the first day are proteins. So how are proteins made? Well, proteins are made by information in our genes. And the way to understand what genes are made of DNA. And notice that genes are made of 2 different strands that come together in a double helix with 4 types of building blocks, which very specifically will link only with a specific type of base. Now, that's shown in a cartoon way as these pegs, which will only fit in, this sort of V shape won't fit with any other type of peg except a green one. And similarly, the yellow peg can only fit with a red peg. But if you want to look at it in chemical detail, that's shown here. This is what these pegs really look like. They're these chemical groups, and this fitting is really what we call base pairing. That is to say, a very specific type of interaction so that a G will only recognize a C. These groups are such that they will only recognize a C. They will not recognize an A or T and still fit inside that helix. And notice that the helix is not simple, but it consists of an alternating arrangement of sugar and phosphate that are linked up, and each of the sugars then is linked up to 4 types of bases. Now, the fact is that these bases are occurring in a specific order. You can think of these bases as letters in an alphabet, and you can think of them as forming a sentence which consists of a sequence of letters but using a 4-letter alphabet. Now, that's what our DNA consists of, and it consists of 2 strands. Now, imagine if you take one of those strands and make a copy of it into a molecule called RNA. This is called messenger RNA because it carries the message from the gene. But this messenger RNA is very similar in that it consists of the sugar, phosphate, etc. So it's a polymer that's very much like DNA. There's a subtle but very important difference. But it has the same sequence of bases, okay? And there are 4 types of bases. But remember I told you that proteins are chains of amino acids. Well, they're a completely different type of polymer. They're called linked up by peptide bonds. Notice that there's no sugar or phosphate here. And they consist of 4 different types of building blocks, 20 different types of building blocks based on these chemical groups that are attached to each of these amino acids. So what you have to think of this is a sentence but made up of a 20-letter alphabet. And you have to go from this 4-letter alphabet to this 20-letter alphabet. And then this polymer, when it's made, will fold up into all of those variety of shapes. And depending on the exact sequence, it'll fold up into a particular shape, which gives it a particular function. And that's why some proteins carry oxygen, other proteins form the skin, other proteins are antibodies, and other proteins detect light or conduct potassium through our membranes and so on. So how does that all work? Well, it was noticed early in the 50s that if you made newly synthesized proteins in cells, they would localize on these blobs in cells. And when people isolated these blobs, it turned out that they were particles which consisted themselves of RNA and protein. So although they were making proteins, they themselves were made of proteins. This is a paradox I'll come to later. But these particles consist, as you can see, of 2 subunits, a large subunit and a small subunit. And that's true in all organisms. And they were called the ribosome because they were made of both RNA, which is ribonucleic acid, as well as protein. And because they were seen first as particles in the cell, and they were discovered by cell biologists and not by biochemists, they were given the name some, because some is a word for particle. So this is the ribosome from bacteria. You can see that the ribosome itself is a highly complex molecule which is made up of smaller entities. So it's made up of many different proteins and large pieces of RNA. It's about 2/3 RNA by mass, but the whole assembly consists of about 50 different proteins that make up the 2 subunits that are brought together, and this is what it looks like. Now, how would it work? Well, it turns out that there's an adapter molecule which brings along an amino acid, and the ribosome has slots for these adapter molecules. These adapter molecules are called tRNA. They themselves are made up of RNA, And they can base pair, just like those bases can base pair specifically in the two strands of a helix. These three bases here can base pair with these three bases on the mRNA, which carries the genetic message, which is that sequence of bases that contains the genetic information on how to make a protein. And what the ribosome has to do is read this information and stitch together a protein. And so here is the first tRNA bringing in the first amino acid. And then what you see is that the second tRNA comes in, and only if it is the correct match, that is, only if this pairing is correct, will the ribosome link up these two amino acids. If it's not, the tRNA will leave, and then other tRNAs will come in until the right one has entered the ribosome, and then the two tRNAs will link up. And that's why it's synthesizing these according to the specification in the gene, okay? And then you notice that the ribosome has to move, and it does that in two steps. And now the third triplet of bases is exposed in that site, and so the third tRNA can come in, and again, it has to be the correct one, otherwise it will leave. And if it's the correct one, then the pairing, the bond between the second and third amino acid is done. So notice what's happening here. It's reading along groups of three bases here, and according to the specification, it's making a linkage between amino acids at the other end. And so by the time it's all done, you've read the gene and you've made a protein polymer, a polymeric chain of amino acids, and then the protein is made. Now, of course, this is a simplified cartoon. Remember I told you about representing molecules. And this tRNA, of course, doesn't look anything like this. In fact, it was discovered its structure was determined in the mid-1970s, and that's roughly what it looks like. Even this is a simplified representation, but here is the triplet of bases that pairs with the triplet of bases on the genetic message. And if you want a more realistic representation, here's what it looks like. This is a view that was first seen by cryo-electron microscopy, a technique that's becoming increasingly useful. And you can see that this is where the genetic message binds. It binds to the small subunit, and those groups of three bases that I showed you that interact with the tRNA bases are here and here. This tRNA holds the growing protein chain of amino acids. So it's a linkage of amino acids. And then here is the new amino acid that's brought in, which has to match this triplet. And if it's the correct match, a bond will be formed between this growing chain and the new amino acid. So the chain will be longer by one amino acid. And then the whole thing will move, and the amino acid will go on making protein. The ribosome will go on making protein. So antibiotics somehow block this process. Now, if you really want to understand how antibiotics block this process, you need to understand what this machine looks like in atomic detail, and then you need to understand exactly how antibiotics interact with that atomic structure. So that leads to the question of how would you determine what an atomic structure looks like. And what you would do if you wanted to look at an object in great detail is you would take an object and you would put a lens in front of it and magnify it so that you could see the details of the object. Now, what is a lens actually doing? What a lens is doing is gathering rays that are scattered from the object and then recombining those rays in order to form an image. Every one of you at this moment is collecting scattered rays from the screen, and then the lens in your eye is recombining those rays to form an image in your retina. Now, that image can be much smaller than the object, as in the case of the image in your retina. Your retina is only about that small, and the screen is enormous. So for a telescope or your eye, you're actually seeing a reduced image. But if you have a magnifying glass or a microscope, you can see an incredibly magnified image. Unfortunately, there's a rule in physics which says that you cannot see details that are smaller than the wavelength of the light that you're using. Now, light has a wavelength, and we've seen that in other talks, of somewhere between 400 and 700 nanometers, okay? And the distance between atoms is about 0.2 nanometers, or about 2 angstroms. So light is many thousands of times bigger than the distance between atoms. So there's no way you can use visible light to look at atomic structure. The way you do that is by using radiation, light radiation, and it was, as we learned, it was James Clerk Maxwell who showed that light is actually a form of electromagnetic radiation. Now, if you take electromagnetic radiation of the wavelength short enough to see atomic details, that's radiation of photons of 1 or 2 angstroms in wavelength. That's what we call X-rays, okay? Now, the way to solve structures with X-rays is similar to a lens. Now, I won't go into details, but if you were to shine X-rays at a single object, there wouldn't be enough signal in order, even if you were able to focus it, there wouldn't be enough signal if you shone very powerful X-rays to get enough signal from the object. You would destroy it before you even had signals, because X-rays can actually damage the object they're looking at, unlike ordinary light rays. And this is why if a dentist takes X-rays of your teeth, they're very careful to put a lead apron on themselves, because they don't want to be exposed to X-rays. And even the patient is exposed only to a tiny dose of X-rays. But in order to amplify the signal from X-rays, you could do that if you crystallized your object. If you crystallized your object, then you're adding up the signal from millions of molecules. And so you're getting millions of times the signal you would get from a single molecule. That's one of the advantages of using crystals. The other advantage of using crystals is that the way you combine rays is you're combining waves. And in order to combine waves, you not only need to know how strong the wave is, that is, how high the troughs and crests are of a wave, but you need to know from different parts of the scattered radiation how far advanced or behind each wave is. That is to say, each wave will not have a crest that occurs at exactly the same time as the other waves. And you need to know how far ahead or behind they are in order to be able to combine them. Now, lens does that without thinking about it, but if we're to do that, we need that information. And you can only get that by using crystallography. And this was a technique that was developed by Max Perutz and his former student, John Kendrew, who became his collaborator, for which they received the Nobel Prize in 1962. And so the way you do that is you take your crystal and you hit it with X-rays, and you take the scattered rays, and if they're crystals, they will only scatter in certain directions. If it's light rays without a crystal like the screen, it goes everywhere along in the room. It doesn't matter where you're sitting. But if it's a crystal, you would have to sit only in certain locations. Otherwise, you would see absolutely nothing. So what you see then are these scattered rays which are concentrated into these spots. And then you do in a computer what a lens does, which is recombine them to form an image. And that's how you would get an atomic structure of a molecule. And this is a a method that we're celebrating the 100th anniversary of, because it was first discovered, the fact that X-rays are waves was discovered by von Laue in 1912, in the spring of 1912. And then in November of 1912, a 22-year-old PhD student named Lawrence Bragg showed that these X-rays would be concentrated in certain spots and that you could actually determine structures from them. The following year, he, in collaboration with his father, but he actually had some of the key ideas, went on to determine the structure of simple molecules like common salt and various minerals. And then 3 years later, he went on to win the Nobel Prize at the age of 25, which is still the record for Nobel Prizes. And just 2 weeks ago, I gave a talk in Cambridge to celebrate the 100th anniversary of Bragg's equation. So we need, for these things, crystals. And Ada Jonat, working with Heinz-Günther Wittmann, obtained the first crystals of the 50s subunit, which was a remarkable achievement because the 50s subunit is about 1.5 million Daltons in molecular weight. It was about 10 times larger than anything that had been, maybe 5 times larger than F1 ATPase, which had been crystallized then. And then a few years later, a group in Russia crystallized both the small subunit and the large subunit. Now, these crystals were not sufficiently well ordered, by which I mean crystals are three-dimensional arrangements of molecules. So for them to be well ordered, each molecule has to be in exactly the same orientation. Otherwise, the atomic information gets blurred because you're averaging molecules which are in slightly different orientations. So these crystals were not of a particularly good quality, and that took a long time. Now, the other thing was that these crystals, because they had a lot of water in the crystals and they're of very large molecules, in order to get information, you had to hit them very hard with very high intensity of X-rays, and they would start dying in the beam. They would start being damaged in the beam. And so a technique called cryocrystallography, which is used to cool the crystals to liquid nitrogen temperature, also had to be used. And then, after investigating many other species, Ada Jonat's group showed that you could get 50S crystals that could be of sufficient quality to maybe solve the atomic structure. But then it was a long struggle, and the struggle was to understand how far advanced or behind each of those waves and those spots are. It's called the phase problem, and that was solved much later. And in fact, these 50S structure was really solved by Tom Stites and Peter Moore and their colleagues at Yale, for which Tom Stites justifiably shared the Nobel Prize. Now, in addition to crystals, you need very powerful sources of X-rays, and here are some of the powerful sources that we use. These are called synchrotrons, and they're these large particle accelerators which provide very powerful sources of X-rays. And recently, we've often used the Swiss light source, and I have to tell you that I've occasionally flown into Zurich airport and gone to the train ticket window, and I'd say, I want a round-trip ticket to Willigen, please. And she would say, oh, you're going to the PSI, which is the Paul Scherrer Institute, where the Swiss light source is. And I said, well, how did you know? And she said, why else would a non-Swiss want to go to Willigen? Okay. Anyway, so as you can see, we've used synchrotrons from all over the world. So the US Navy has a famous saying, which is join the Navy and see the world. And of course, it's a complete lie, because these poor young sailors are roped in, and then they only see the inside of a ship under terrible conditions, okay? And so my people are also cheated because they only see the world's synchrotrons, okay? And they don't see Chicago, and they don't go skiing in Grenoble, you know? They actually, most of the 24 hours a day, this is what they see, okay? And this is the inside of a synchrotron, and you can see this is where the X-rays come in from the ring. So the ring is over here where the particles are accelerated, and then the X-rays come out. This is where the crystal is. And this is a device that has a stream of very cold nitrogen gas at about 100 degrees Kelvin that keeps the temperature cold. And then the X-rays are scattered, and then they're detected in what still is largely a CCD detector, although a new technology developed by people who made detectors for CERN and then later X-ray detectors for the PSI is being increasingly used. But a lot of our data was obtained on CCD detectors, and it's a slight paradox that that in 2009, the Physics Nobel Prize was shared by two people who, for their invention of CCD detectors. And so the chemistry winners were able to thank the physics winners for making their own prize possible. Okay. So anyway, that's how the experiment is done. When the experiment works, which is not always and not, in fact, not usually, you end up with an image of the object. The image of the object is not the atomic structure of the object. It's simply what the object looks like. And you have to then interpret it in order to get an atomic structure. So what you get is something that looks like this. And you know, it doesn't tell you where the atoms are. And you have to figure out where the atoms are from this. And this is a bit like solving a complicated three-dimensional jigsaw puzzle, okay? But it has some important differences from a jigsaw puzzle. One is it's a real experiment. Real experiments have errors. The equivalent, the analog for a jigsaw puzzle is, imagine that people made a mistake when making the puzzle at the factory, and they mixed up some pieces that come from some other puzzle that don't really belong in your puzzle. And there are also pieces from your puzzle that are missing, okay? That's what a real image would look like. There are pieces of image that really shouldn't be there because there are errors in your experiment, and there are other pieces that are missing, either because the molecule is not static, it's moving around, so it's blurred and so you don't see it, or because of mistakes. The other problem is that it's in 3 dimensions rather than in 2 dimensions like a jigsaw puzzle. But of course, we all know the biggest difference is the most important one. When you go to buy a jigsaw puzzle, they very kindly provide you the answer on the cover of the box. So you already know what it should look like, okay? But in an experiment like this, we don't know what the object looks like. So how do we go about it? Well, how do you go solving a jigsaw puzzle? Those of you who like to solve jigsaw puzzles know that you start, first of all, separating out all the ones with straight edges and the corners, and then you build the corners and edges first because you can recognize them easily. Then you start building inward, but you start building parts that you can easily recognize first and save the complicated ones like sky or grass and so on for the very end, okay? And that's exactly what we do for determining structures. You look around and you see some pattern that you can recognize. And here you see there's a regular chain here where you can see these bumps that are connected by these little linkages, and then there are little protrusions. And interestingly, on the other side, you see another little feature, again with regular bumps with little features that are connected. And if you know what you're doing, then you recognize this as a piece of double-stranded RNA, or double helical RNA. So here's the sugar, phosphate, and so on. And those are the bases. So you can see now that you've filled what we call the electron density or the image with our interpretation of what is making that image, which is the atomic structure. And so there you are on the other side. So now what you've done is you've converted a little piece of the image into an atomic structure. You've interpreted it in terms of an atomic structure. Now what you have to do is figure out where is this little piece in the overall object. And you do that and you realize that it's in some corner of the object, and then you start building the rest of the object using the technique I told you until you have accounted for the entire image. There's nothing else left to build. And when you've done that, you've got the atomic structure of the object from your experiment. Okay? You can, of course, make mistakes. Sometimes you can make terrible mistakes, and people do, and then they have to embarrassingly retract their paper and say, no, that's not the real structure. The real structure is this. That's when you do this wrong, but there are many tests that we can do to make sure that our structures are correct. Using this, in 2000, the structure of both the large subunit and the small subunit of the ribosome were determined. And one of the interesting things is, remember the ribosome has to bind the genetic message, and it has to bind those tRNA molecules that have to recognize the genetic code and also bring along the amino acid. And then the ribosome has to stitch together two amino acids, or the amino acid with the growing chain. And one of the interesting things was that this is where the amino acids are linked up. And this is where the genetic code is recognized. And what we found was, when these structures came out, was that those parts, which are the crucial parts of the ribosome, were made up only of RNA. Now remember, there are three kinds of RNA I'm talking about. One is messenger RNA, which is a copy of DNA, which contains the genetic message, which is the sequence of information used to make a protein. The other is tRNA, which is the connection between the amino acid and the group of three bases on the genetic message that it codes for. That's the small adapter molecule that the ribosome binds to. And then there's ribosomal RNA. Remember, 2/3 of the ribosome itself is made of RNA. So suddenly we're seeing that in the machinery that makes proteins, RNA is what's important. Okay? There's RNA in all of the players in the ribosome. And the ribosome itself consists of RNA, 2/3 of it. This solves a paradox, which was first proposed by Crick before he had any idea that RNA could do things. He said that maybe ribosomes consist today of both proteins and RNA, but then that begs the question of where did the first ribosomes come from if they themselves are made of proteins and they're used to make proteins? It's a chicken or egg problem. And he said maybe the early ribosomes consisted only of RNA. At that time, people did not realize that RNA can not only carry information like the sequence of bases, which can do base pairing, but can also do chemical reactions. They can carry out chemical reactions like proteins can. But in the 80s, it was discovered that RNA can also do catalysis. It can carry out chemical reactions. So the same molecule can both carry information and do chemical reactions. And so people now believe that life began as an early RNA world, which could not only reproduce itself because it can carry information, but it could carry out reactions. But it's not as efficient as proteins. But once the ribosome evolved, then it started to make proteins, and then proteins quickly took over and started carrying out most of the reactions in the cell. So it's a glimpse into an early RNA world. Now this is what the whole ribosome looks like. That was done first for the empty ribosome in 2005, and then later, a year later in 2006. And this shows you the two halves of the ribosome. in magenta or pink is shown a little piece of the genetic message, and in green and red are the tRNA molecules. At one end, they're reading the genetic message, and at the other end here, they're connecting the amino acids together to make the protein. And just to give you a slightly different view, this is a different view of the ribosome, and I'll rotate it to give you what's a classical view of the ribosome with the large subunit and the small subunit and the tRNA molecules nestled in between. So I've told you a lot about the ribosome, but I started off with infectious disease and antibiotics. So now that we have atomic structures of the ribosome, it was then possible to determine exactly how antibiotics bind to it. And the way you do that experiment is you take your crystal with the antibiotic and then solve the image with the antibiotic. And you subtract away the image from the ribosome itself. And what's left over is just the image of the antibiotic, which is this little blob here. And this you can fit the antibiotic into that image. This particular antibiotic is called spectinomycin. Normally you wouldn't want to take it, but it's still useful if other antibiotics don't work. And you can see that this tiny molecule actually binds here in a tiny cleft in this ribosome, and the ribosome has to move. I'll show you much later some of these movements. And what this antibiotic is doing is probably preventing an important movement, and that's why it works. Now, another antibiotic, remember, these tRNA molecules which are bringing in the amino acid have to make a base pair interaction with the group of 3 bases on the genetic message. So another antibiotic called tetracycline works by stopping that interaction, by blocking that interaction. How does it do that? Well, here's the genetic message, and here is tetracycline, okay, right here. And the way it works is this is where the tRNA binds, and this is where the base pairing would occur. And in red is shown where tetracycline binds. So if you have tetracycline, then this tRNA molecule cannot come in because it would clash with the tetracycline molecule. And so you would not be able to add to the protein chain when you had tetracycline. So that's a simple way of doing it. Now, a large number of antibiotics bind by binding either here or here. It turns out that the proteins are made here, but the amino acids are joined together here. That's called the catalytic center or the peptidyl transferase center. And there's an antibiotic here called chloramphenicol, which binds right there. And then this is a tunnel through which the newly made protein has to come out of the ribosome. And there are antibiotics which work by binding at the beginning of the tunnel, so they won't let the protein go through, okay? So I'll show you that in a couple of ways. So here is the ribosome. Again, these are the 2 tRNAs. This tRNA has the growing protein chain, and this tRNA brings the new amino acid. Where are they linked up? We can zoom in on the area where they're linked up, and what you see is the center, the catalytic center, where these 2 amino acids have to be joined together to make a protein chain, okay? And one of the antibiotics, called chloramphenicol, binds exactly where one of these amino acids would be. And so if that antibiotic is present, then the amino acid cannot enter the pocket in order to be joined. And so it stops amino acids from being joined together. Now, other amino acids bind near chloramphenicol. So this is chloramphenicol. These are called macrolide antibiotics. For example, erythromycin or azithromycin, which are still in very common use today. And just to show you what they have in common, they have this ring structure, which is called the macrolide ring. And you can see they don't bind where chloramphenicol binds. They bind rather at the entrance to the tunnel, but not too far away. Notice they bind fairly closely together. So when these structures were determined, Tom Stites and Peter Moore and their colleagues founded a company called Ribex Pharmaceuticals in New Haven. And the idea of this company was to The idea of this company was to use these ribosome structures and to use the knowledge of how antibiotics bind to maybe design new antibiotics that might be more effective. Now, I'll show you some work from that company, and then I'll have some comments to make about it. This is chloramphenicol, and this is where azithromycin binds, okay? Notice that they're binding next to each other. Now, there are problems with both antibiotics. I may be the only person in this room who has taken chloramphenicol, because in the 50s and early 60s, it would be used for typhoid, okay, which is often common in developing countries, but it's not a nice antibiotic. It's highly toxic, it has side effects and so on. So there are other antibiotics which are now used. So chloramphenicol is not used because of its side effects. On the other hand, macrolides, there are many antibiotics that are resistant, many bacteria which are resistant to macrolides. For instance, here are some MRSA strains and Streptococcus pneumoniae strains. These are all strains. Red means that you require such a high concentration that is effectively useless, okay? So these are all resistant strains of to macrolides. So you have two antibiotics which bind nearby, both of which have problems, okay? But what the people at Ribex said is, we know their binding site, and now we know that they're binding right next to each other. Could we design a molecule which binds to both sites at once, okay? And they designed a compound called RX-2102. Now, this is a very large molecule, so it would bind very specifically if it works, and therefore it would avoid some of the side effects of chloramphenicol, and it would also avoid if it's resistant here, they can make it so that it overcomes the resistance because they know what the resistance is due to. And they went ahead and synthesized a compound, and then of course they have to check if their ideas are right, so they went and solved the structure, and in fact the molecule bound exactly as they had predicted. And lo and behold, it's effective against all of those resistant strains of molecules. Now, there are not just macrolides and chloramphenicol, there are other classes of antibiotics, lincosamides and oxazolidinones and sparsomycin. You can see they're all binding in this space next to each other. So by combination, you could make all kinds of new molecules that occupy more than one space, perhaps even two spaces and so on. It would depend on the chemistry and so on. Now, interestingly, Ribex went through many rounds of venture capital funding, many more than is typical. And then, this is not a secret because you can look it up on the web, it tried to launch an IPO last spring. And the IPO was withdrawn because the share price that they could get on the market was a lot less than what the investors had put into the company. And this at about the same time, a company whose main benefit is that you can tell your friends what sort of perfume or movies you like, or what pizza place to go to, etc., was worth billions of hundreds of maybe $100 billion, okay? And so of course it's worth about half of what they thought it was worth, but nevertheless, it shows you that what's essentially an advertising medium is worth a huge amount of money, and something that saves lives is not, worth, as much as what investors had put into it. And this means that the current way of making pharmaceuticals is not really working, okay? We need to think about how pharmaceuticals, new medicines can be made. Because pharmaceutical companies don't really like infectious, development of antibiotics. Many companies have withdrawn completely from antibiotic development because you give antibiotics to a patient for a week and they're cured, okay? And you don't make much money off that. What you want is a pill that the patient has to take all their life, The ideal patient is someone who's, has hypertension, high cholesterol, maybe depression, impotence, whatever, and that they have to keep on taking pills all their life, okay? And so that's a serious problem. It costs $1 billion to develop a new drug, but one thing that we can guarantee, and we know because of Darwin why that's so, is that whatever you make, resistance will always emerge. And for instance, in bacteria, I've told you antibiotics depend on a tight fit and precise fit between the antibiotic molecule and the pocket where the antibiotic is binding. And if you can destroy that fit, then you can destroy the effectiveness of the antibiotic. And you can do that in many ways. For instance, cells make enzymes that break down the antibiotic. So now the parts are much smaller, so the fit isn't tight, and so it doesn't work. Or they can add little extra groups to the antibiotic, and so now the antibiotic has the wrong shape and doesn't fit into the pocket. Or cells actually make pumps that pump antibiotics out of the cell. So again, it's useless. So the point is that whatever you do, resistance will emerge. And so infectious disease has to be controlled in many ways. One is you have to promote the rational use of antibiotics. You have to make sure that you have good surveillance so you can see when epidemics emerge so you can control them. You have to have good infection control, which means isolation as well as sanitation measures. In England, the number of hospital deaths from MRSA dropped dramatically when the NHS imposed a rule that everybody entering the hospital and entering a ward had to wash their hands. No matter how often they went in and out, every single time they had to wash their hands. That alone resulted in a dramatic reduction of deaths. And then finally, this is where scientists can help. That is, they can understand why some bacteria cause disease and not others. That's microbial pathogenesis. They can have better diagnostics for bacteria and disease to catch them early on. They can have vaccines. That's, again, a big field. And then the part that I talked to you about, which is based on molecular structures, is development of new drugs and new therapies. I want to close by pointing out how complicated the ribosome is. And before I'll show you a movie of how the ribosome works in sort of broad detail. We're still working out all of the details now, and that's going to take years still. And certainly, if we want to understand ribosomes from humans, it's not going to be done in my lifetime. But before I tell you the movie, I want you to think about this. Every single molecule in every one of your cells was either made by the ribosome or made by enzymes which were made by the ribosome. So really, this is an ancient fundamental molecule that predates life as we know it today. Okay? So I want to show you the movie of the ribosome and close with that. So this long molecule is the genetic message, the RNA containing the genetic message. And the small subunit, first of all, has to know where to start on that sentence. And that's done by the help of these three accessory factors And in blue is shown the first tRNA, bringing the first amino acid. And then the large subunit joins, and you're left with the genetic message and the first tRNA, which has the first amino acid. And now the next tRNA has to come in, and that too is brought along by what is called a helper protein or a factor, and is delivered to the ribosome. Here is where the match is recognized, and if it's correct, the tRNA is allowed to move in, and then the bond is formed between the first and the second amino acid. Then the two tRNAs have to move, and that happens in two steps. First with the large subunit, and then with the help of a protein factor, the thing moves with respect to the small subunit. And notice it's dragged the message also with it, so that now the next group of three bases is going to be recognized by the third tRNA, which is bringing in the third amino acid. This is all something I showed you in the cartoon form, but now this is what has to happen as it's going along the gene, reading it, and it's much faster than this. But if I showed you that, it would be a blur. And the protein chain comes out of the tunnel and then folds up. The information to make the shape of the protein is actually contained in the sequence of the protein itself. And often proteins will fold up into their shape by themselves, but sometimes they require helper proteins in order to make them shape. Then it comes to the end where a stop codon is recognized. A special signal called stop is recognized by a factor which cleaves the protein away so it can go off and do its thing. And then other factors come in and split the ribosome apart so the whole process can start all over again. This is the rough outline in bacteria, and in humans, the way that ribosomes start is much more complicated. Other parts are very similar to what I've shown you. So I'd like to thank you for your attention, and I've had a great time.
Thank you for that wonderful talk, which is actually the fifth wonderful talk this week. We now have time for questions, and so if you have questions, you have the cards which were distributed when you came in. Please write your question either in French or in English, and the hostesses will pick them up and bring them, and the speakers will come up on the rostrum to answer them in just a few minutes. Well, we have many questions, as usual, and very good ones. How long does it take a ribosome to make a protein? How do the tRNAs know they should come close to the ribosome? And how long ago did the first ribosome appear?
So people have measured the rate of making proteins in bacteria, and they're made at the rate of about 20 amino acids per second, okay? And the ribosome only makes about 1 in 1,000 mistakes, okay? So only 1 out of 1,000 amino acids added is actually a mistake. So it's an extremely accurate and extremely fast machine. You can calculate how long it would take to make a protein. It'll depend on the size of the protein, but typically, let's say if you have 200 amino acids, it would take about 10 seconds to make a protein. The tRNAs don't know, when to enter. They don't know that it's the right codon there waiting for them. So all movies are a bit of fiction. There's a bit of Hollywood in that movie, and the reality is that tRNAs are constantly coming in and out of the ribosome. There are many different types of tRNA, and most of them are the wrong one for that particular group of 3 bases. And it's only when they're the correct tRNA for the group of 3 bases that they actually induce a change in the ribosome that causes that factor to do something to let go of the tRNA so it can enter the pocket where the peptide bond has to be, the amino acid has to be added. Sorry, what was the last part?
How long ago did the ribosome first appear?
Well, I don't have an exact figure, but it certainly dates back to a world before there were any proteins. So it's older than when bacteria and archaea and higher organisms diverged, so well over 1 billion years old.
Okay. Another question, could you please comment on the fact that cows and maybe other domestic animals receive antibiotics in their daily feed? What effect on us and on them?
Yeah, so I think this is a terrible misuse of antibiotics. I personally also think it's somewhat inhumane, okay? And I think the idea of giving antibiotics to cows and pigs and so on to fatten them up is a classic example of agriculture as business rather than, and it's a way of using antibiotics in an indiscriminate way that can propagate resistance, because you will then have resistant strains, and then these resistant strains can transmit the information on how to be resistant to other strains which can be pathogenic and thereby help to spread. So for all sorts of reasons, I think this is a bad use of antibiotics.
Somebody else, I'd like to know your opinion about vaccine and new drug therapies. Which way do you think is better?
Well, I mean, if we can have vaccines, obviously it'd be better, because first of all, prevention is always better than cure. Sometimes you may get the disease in such a terrible way that maybe even with antibiotics you might die, okay? So I think it's always better to have an effective vaccine. It's not a competition between the two. They serve very different functions. The problem is that many bacteria, for reasons we don't understand, do not mount. You cannot make a good vaccine against them. And people are working very hard to understand what it is about some bacteria that allow them to make a vaccine against them and others not. I'm not an immunologist, so I can't comment on the technical aspects, but I know that this is a huge problem that many people are working on.
There are two sort of linked questions. One, it takes the negative view. If bacteria develop resistance, is it possible that a bacteria becomes resistant to everything? And another person asks, do you think one day you'd make an antibiotic which no microbe could resist?
Yeah, so let me take the second one first. I believe that natural selection is so powerful, the idea of mutations that can get selected for under selective pressure, it's so powerful that, it's powerful enough to have created humans from the earliest little blobs, that existed on Earth, right? So that shows you the power of natural selection. But it's so powerful that resistance will always emerge. There will always be ways that bacteria will find a way around, things. Some antibiotics could delay it. They may be more difficult to acquire resistance against than others because they're hitting centers that cannot be mutated very easily. But my belief is that it'll eventually emerge. The flip side is, are there bacteria that can emerge that will be resistant to every known antibiotic? We may be very close to that today, okay? There are some strains of Staph aureus that are resistant only to, sensitive only to one antibiotic, which is vancomycin. there are other bacteria which are resistant to vancomycin. If these two were to cross, you could get a superbug that's resistant to everything. In India, last January, when I was actually in Bombay at the time, I opened the newspaper that morning to read that there were 7 cases of what's called TDR-TB, totally drug-resistant TB. This was TB, an outbreak in Bombay, which was resistant to every known TB antibiotic. Okay, so this is actually a serious problem, and it's why we need to not use antibiotics indiscriminately. It's very, very difficult to make a new antibiotic. Not only is it $1 billion, but you know, this molecule I showed you, we don't know what its side effects could be, this new molecule that they synthesized. All we know is that it'll kill resistant bacteria, but we don't know what will happen if you put it into humans. Will it cause side effects? Is it toxic? Is it used efficiently? No, you know, those are all big problems. So it's very, very difficult to make a new antibiotic. It's easy to destroy a useful antibiotic by indiscriminate use. And so that's one thing. The other thing is, when resistance emerges, maybe what we need to do is have a ban on that particular antibiotic, that is to say, rotate it out of use. Then what will happen is, just by natural selection, you know, in order to become resistant, the bacterium has to make something extra to be resistant. That costs something. And so if you don't have the antibiotic, you don't have that pressure, then the bacteria will slowly lose the resistance in the general population. Then maybe 10 years later, that antibiotic can be useful again. So, but this requires social coordination between public health officials in different countries and so on. So that's a social problem, not a scientific problem.
Here's a general question. Are these mechanisms universal to all life forms on Earth?
Are these what?
Are these mechanisms universal to all life forms on Earth?
Yeah, so ribosomes, as I told you, because they make proteins, are essential to all life forms. As I told you, every single molecule in every one of your cells, including all of the bacteria in your body, who far outnumber your human cells, by the way, all of them make proteins, okay? And they're all made by ribosomes. And even if viruses invade a host cell, it uses the host ribosomes to make proteins. So you can't have any proteins without ribosomes. And the mechanisms are highly conserved. Now, having said that, there are important distinctions. Ribosomes from higher organisms are slightly different. Otherwise, antibiotics would kill us too. They work by binding to the pocket's shape of the pocket is slightly different for the antibiotics that are useful. There are many antibiotics that are not useful because the pockets are the same. And the mechanisms are largely the same, but the way that they learn where to start on the genetic message is very different in higher organisms like humans compared to bacteria.
A little bit in the same line, there's a question here saying, do the antibiotics interact only with one site? Can the antibiotic interact with other sites, and how will this change things? How can you prove it?
Mainly they work by binding to a site that is crucial for the ribosome's function, okay? It destroys some crucial aspect of this machine. The ribosome, if you think of it as a machine, has all these moving parts and things have to fit in just the right way. And if you destroy that movement or function, then that's how they work. And so they work by binding to a precise site. Now, there are antibiotics which bind to more than one site on the ribosome. We've found that in our own lab. But we don't know if these other sites are actually meaningful, because if you add antibiotics at very high concentrations to a crystal, sometimes they'll even bind in what we call non-specifically. That is to say, just by accident, they bind somewhere else. But that's not really how they work. The place where they bind specifically is usually just one site.
We'll give you a rest. There's a question for Dave Lee here. we saw your molecules moving. The question is, objects in the macro world are static, objects in the nano world are constantly moving because of Brownian movement. How big does an object have to be not to be influenced by Brownian motion?
So that's a very good question. It's actually a continuum, as you might expect. So objects in the macroscopic world, although they don't appear to move, the atoms in those objects are actually still moving. It's just that the movements are of the atomic scale, and so they don't have any visible effect to you when you're looking at them. So if you're down at the nano world, a millionth of a millimeter, everything's in constant motion, and those effects dominate. If you go up to about the micron level, fluctuations are still dominating, and you can are still going to be affecting all aspects of motion. If you go up to the millimeter scale, then they sort of stop, and everything will look the same. So it's somewhere between, I guess, millimeter scale and right down to microns that Brownian motion begins to become effective. And in fact, it was first observed by this phenomenon of Brownian motion, as the name suggests, by a Scottish botanist, Brown, looking through his microscope at some pollen grains, and he noticed that they were actually in constant movement, and that was the first time that the phenomenon was discovered.
It will change slightly because somebody asked, a lot of research groups are interested to bind different molecules to DNA. Could you comment why? Anybody else?
Sorry, different?
A lot of interested to bind different molecules to DNA, and the binding to DNA. Why are people trying to get molecules that bind to DNA?
Why are they finding
Or
molecules?
a lot of research groups are interested to bind different molecules to DNA.
The reason is that our genes reside on DNA, and they contain information to make proteins. But they contain much more than the information on how to make the protein. They also contain information on where to make it, when to make it, and in fact, sometimes how much of it to make. Okay? And this is called control of gene expression. That is, you're controlling how much of the gene is expressed, that is to say, converted to a protein. And that itself is done, that control is done by proteins that bind to signals at the start of a gene. And so if you want to control whether a cell is dividing too fast or too slowly, or if you want to make more of something or less of something, then you have to find proteins that will bind to that particular part of DNA that controls that particular gene. And that's the big interest. Because, for instance, diseases like cancer are where the control of gene regulation has been lost. It's gone out of control. Things which should normally be controlled are not in control, and you may want to regain control. In other cases, you may have diseases or some problem where something that is supposed to be made is not being made, and you may want to turn on something. So that's the interest.
Somebody here has a question. What is your opinion about genetically modified organisms? Is there a risk in eating meat of animals that have consumed GMO? You've got round the second problem.
Yeah, so I think, any new technology can have some dangers that can be exaggerated. My feeling is no one who rides a bicycle on a public street or drives a car should be worried about GM crops, because you have far greater dangers in your life, okay? And the problem is that people when a technology is new, it's often possible to scare the public, okay, by creating Frankenstein type of images. And the fact is, we've been genetically selecting plants and animals for centuries, okay? Every species of cow that roams on the hills of Switzerland has been selected for over centuries for either optimal beef or optimal milk production or whatever. All of the strains of wheat have been selected. We don't eat the wild wheat that our forefathers, in prehistoric times ate. The yield, they've all been carefully chosen genetically for high yields and resistance to weather and so on. So, and then people always compare some theoretical danger to, to some and don't consider practical benefits. Now, I'll give you an example. One of the ways that you can use genetically modified crops is to have the plant produce a pesticide, so it'll be pesticide resistant, okay? And some people think, oh, this is terrible because, what if this then escapes, etc. Well, that would be fine in some artificial universe and parallel universe that doesn't exist. The real universe is that you could use that, or you can use chemical pesticides, which can run off into your drinking water and pollute, your streams and lakes and so on. Which would you rather have? Okay, I would rather have a plant where the pesticide is made inside the plant and, can resist insects. Incidentally, those pesticides that the plant make are highly specific towards certain species of insects. You can eat those plants and nothing will happen to us, okay? Now, the other thing is, none of this happens in a vacuum. Any new technology is highly regulated. All of us who do experiments, I answered an ethics question the other day, all of us who do experiments in labs have tight regulations. And a country like Switzerland has extremely tight regulations. And so all of these crops have to be tested. They have to be tested inside a laboratory first in a closed environment, and then in a field where it's highly constrained, and there are methods so that the plant maybe needs some specific nutrient, or it can only survive one generation. There are all kinds of safeguards that you can build into. So I think we should use the benefits of genetic modification and be careful about it, okay? And I think humanity can benefit a lot. A classic example is night blindness. There are millions of people in Asia and in developing countries who are going to have night blindness because of vitamin A deficiency. There is, in fact, a genetically modified form of rice which is golden in color because it produces the same beta-carotene that's present in carrots, okay? And if people eat this golden rice, they can get enough vitamin A to prevent night blindness when they're growing up. So who are we in rich countries to tell these people that they can't have the benefits of genetic modification?
Did you want to say something?
Yep. John? I think 75% of canola now is produced by genetically modified organisms. But the one thing that I would ask you to consider is building on your argument that nature is very good at developing resistance. So I think when we have the world's best herbicide, which was called glyphosate, commonly known by the company that made it as Roundup, and then we engineer plants that are resistant to that best herbicide, we're accelerating the process of resistance unnecessarily.
Well, possibly, but I should tell you that these biological insecticides are actually things that other plants have themselves produced in nature. They're not things that humans have invented. They've simply taken a plant that normally resists an insect, found out what it is about that plant that resists the insect, and transferred it to some useful crop, okay?
No, I think it's different.
Yeah, so it is different. Yeah. And Whereas herbicides that we use, which are chemical herbicides, are often indiscriminate. Not only will they kill not one particular type of insect, they'll kill a range of insects. And so they have even greater problems.
Perhaps a general question is, you mentioned the problem that, I imagine you were talking about Facebook, and drug companies have different values in our civilization. What measures, I wonder, could be taken to encourage research in pharmaceuticals against diseases which are still fatal for millions and in some cases likely to become again fatal for millions?
Yeah, this is a big problem because pharmaceutical companies are not in the business of making medicines. Let's be clear, they're in the business of making money, okay? And that's fine, you know, that's what businesses exist for. So there are several approaches. So clearly, you can't expect a pharmaceutical company to spend billions of dollars if it's not going to get a return on its investment. So what are some other models? One is, for instance, there's an orphan drug program where there are diseases that don't affect very many people, and it's not profitable for companies to invest in it unless they're guaranteed a return. And so in some countries, for instance in the US, there are orphan drug programs where the government will guarantee a certain returns to make sure that effective drugs are made against these diseases. There's no reason why this couldn't be done against diseases that affect poor countries where millions are affected. Similarly, foundations, we already know that foundations like the Gates Foundation are spending lots of money on third world diseases. So, that sort of mechanism could be used to encourage research and development for medicines that are not immediately profitable. But I have to tell you the story of penicillin. Penicillin was entirely government funded. No pharmaceutical company would have touched penicillin because of the cost involved, okay? But when people figured out how to make penicillin and figured out how to make it in large commercial quantities, lots of companies made millions of dollars selling penicillin, okay? So it doesn't have to be either or, it could be a win-win situation.
Anybody else got a comment to make? Or, no, wait a minute. Here's a question actually that I asked Rod McKinnon earlier this week, but how could certain geneticians come to speak about junk DNA? Nature doesn't seem to produce senseless things. Is it not just not yet understanding?
The pendulum keeps swinging, okay? When people knew about genes and then they knew about our DNA, they realized that the part that codes for proteins is only a small fraction of the overall DNA. And so they said, well, what's the rest of the DNA doing? And the term junk DNA came into vogue. And the idea is that it's very difficult to get rid of DNA that has entered the genome as long as it's not doing any harm. All of you own, or most of you, I imagine, own computers. And if you go and look on your hard drive, you'll find lots, hundreds, maybe thousands of files, maybe even half of your disk drive, full of files that you have absolutely no use for. Emails that are junk, some file that was a mistake, etc. Why is it that it's accumulating on your disk drive? The reason is it costs energy for you to sit down and clean out your disk drive. It's easier to just live with it, okay? And then, eventually the disk size of your disk drive keeps expanding, and genomes also keep expanding, and so on. There are organisms where the genome cannot expand. Those are viruses because they have to be packaged inside a shell, so there's not enough room for them to expand. And not surprisingly, viral genomes are incredibly compact. They have no junk. In fact, some bacterial viruses use the same piece of DNA to code for more than one gene. They use one strand for one gene, a different strand for a different gene, so they're that efficient. So that's the one way of looking at junk. The other way is that junk actually contains signals. They contain signals on regulating how genes work, how to turn them on and off, and so on. And what people have recently found is that a lot of DNA that we thought of as junk is actually contains signals for making small RNA molecules that can regulate genes, that contain other kinds of information. And so now the pendulum has swung to saying that there's nothing like junk. Well, I don't think this can be true either, because I'll give you the reason that people thought there was junk DNA in the first place. And that is Sidney Brenner pointed out that a species of fish called pufferfish, which is a delicacy in Japan, which is fugu, okay? So this is a highly poisonous fish, and you have to be very careful to remove the poison, or you can die. But anyway, this fish has a very, very small genome, several times smaller than other kinds of fish. Now, this fish can swim around, it can make poisons, it has, prey, it has predators, it's as complex an organism as other fish. And yet it has several times smaller genome. So clearly, the answer is somewhere in between.
Perhaps I could quote Rod McKinnon's reply, which was, he said, "Oh no, junk. He said, the future is junk," he said. Talking about the future, we have maybe in this room the organizer of the 30th Reich Colloquium, which will take place in 2042, I think. Have you, anybody, got any idea what will be a good thing to talk about in terms of molecules then? Just so he can start preparing, or she can start preparing. I realize that, you should never make predictions, certainly not about the future, but is anybody prepared to stick their heads out?
Well, being out at CERN, we have to think about life on other planets.
You think so? Let's go, yeah.
I would like the colloquium to answer this following very simple question, which is, how do we remember somebody's telephone number? Okay? Yep. Because in order to remember somebody's telephone number, you have to have a mental representation of a mathematical entity, which is the number. You have to know it's a phone number. You have to know it's connected to a person whose name has to be stored, but the person is independent of the name. You can just look at the name person's face, and you don't have to remember their name in order to remember their phone number. And all of this is done through the brain, through networks, through molecules, through circuits, and so on. And so a lot of areas, including the area we talked about in this year's colloquium, which is about molecules, have to come together with lots of other disciplines. So,
do you think we'll be using solar energy by then, Andrew?
Oh yes, we're using it already.
but more?
We have to. We have to use the most powerful source of energy that's renewable, at least for the life of the sun.
Materials?
I'm waiting for David's first artificial ribosome.
Okay. I think we could talk on a long time, but they're tired. I would just like to join Jean-Patrick in saying that it's been a wonderful week. When I organized this week, I looked at the speakers and said this should be pretty good, but I was wrong because it's been wonderful, and I'm not personal note, it's been a very great pleasure to have you all here as our guests in Geneva, and we hope you'll visit us again soon. So thank you very much indeed.